Hi, my name is Samuel Gan. I'm from Singapore, and I'm here today to tell you about the Scientific Phone App Lab and our efforts in SpecBT, Gel App, and Colony Counter. I'm speaking on behalf of my team, including Ms. Wu Weiling and Mr. Po Jinjie. Phones have changed significantly in the last 30 years. I'm not old enough to see the one on the top left, but I have definitely drunk from a water bottle like a mobile phone on the top right. And I was extremely proud in 2006 to have my O2 XDA shown at the bottom left. Today phones have changed significantly. They can do much more, and they have sensors that previously was not heard before. And you can even add new sensors to them by the use of Bluetooth, Wi-Fi, and even connectivity devices. What you do see is everyone using the phones for everything but calling. Phones have now been part of the daily life. It's a need in the developed world. Out of a joke, I even took this picture to show that now phones have been can allow people to even do research from even the restroom, right? So this photo won a second prize in one of our A-Stars productivity challenge. Why are we doing all this? Well, many discoveries in the past were made at homes. For example, Alexander Fleming's penicillin, Anthony Leeuwenhoek's microbiology, and many others. Scientists have become now institutionalized in the sense of relying on institutes' research labs. But the recent pandemic lockdowns have shown that scientists need access to the labs throughout. But we've lost the ability to have them at home. But is it a completely lost ability? Well, I strongly believe that it is possible to transform a lab towards the bottom right using the technology and the improvements found in the mobile phones. As a start, it is also my dream to have my lab tech being carried around as a mobile phone with me wherever I go. But I guess I'm a little bit too ahead of my time. It'll take probably another 500 years before this can happen. But let's see, there's still other things that we can do right now. We have a number of efforts on mobile apps that can be used for bioinformatics analysis, lab equipment displacement, even psychological game assessments, and even providing clinical treatment management. All of these apps are free and available on the app stores for both Apple and Android. We've also written 2 reviews back for 4 years ago about how they are changing the way biomedical research can be performed, as well as in some certain cases, clinical and healthcare, and how they can connect doctors and patients and even other features of measurements that patients can do at home. I'll now start with the SpecBT suite. The SpecBT suite was built to bring the spectrophotometer to the samples. In our case, with the power of the mobile phones, we built an Arduino-based device that could send the data from the readings of the cuvettes to the Bluetooth to the mobile phone. This allows field tests to be done. So instead of going to collect water samples from the field and bring it back to the lab 2 hours later where things may have changed, you can now bring a spectrophotometer to the location of test. And in our case, because our bacterial cultures were in a different room from the spectrophotometer, we can now bring the spectrophotometer to the bacterial cultures. We've managed to have this published as an article showing how it can be done. And using a mix of 3D printing as well as Arduino chips, we've made a working spectrophotometer. A video to how SpecBT version 1 is available on YouTube, which you may watch at your leisure. Basically, it just demonstrates how we can connect the phone and the device to turn on and off the light and get the readings. We have compared the readings of our spectrophotometer with that of a real lab photometer, and as you can see, while the absolute values are not the same, they are in the same trend. So this is already a complete prototype. The earlier version of SpecBT is only for visible light. We've made an improved working prototype now, including UV, allowing 260, 280 nanometer measurements of DNA and protein directly. As you can see, there is no cable attached to it, and you can charge it by the port to the battery, allowing the whole device to be completely portable. As with Spec BT1, both are connected to the apps, and the apps will allow editing and also exporting of this data into CSV files. So once you have the Spec BT 1 or 2 CSV files, you can easily plot them on this new app, Stancy. It's a different app from Spec BT app, and so it not only allows importing of CSV files, but also direct entry of data. And you can plot and change the colors of your lines and even the background. And with the screenshot features, you can easily have these pictures for report or some deliverance by social media or even by instant messaging. The advantage, of course, is it's much easier to use than to try and run Microsoft Office or Microsoft Excel specifically on your phone. The next app I'm going to share is the GEL app. Well, it was first created because a lot of students, they do not know how to read the band sizes, even from the marker. And this app can be used for both DNA as well as protein gels. Well, when we first were starting up the lab, we could not afford to buy a gel documentation system. And even though we have one now, it still does not give us the exact band size that we were hoping to know. So based on a few calculations from the distance traveled by the marker, we are able to estimate the band sizes much better. So instead of having an approximation sign in the paper describing that the band size is about this, we can now give a much more accurate band size estimation, and all through the mobile phone. So instead of 5 students queuing up for one gel documentation or 5 researchers queuing up for one gel documentation system, now everyone in the lab has their very own gel documentation system in their pocket, in the mobile phone. To demonstrate how GEL app could improve quantitative biology in band size analysis, we've managed to publish 2 papers using GEL app photos of splice variants. Using GEL app, we were able to give a better approximated number on the gel than simply putting a Wrigley sign and saying approximately 600 or 300 in this case. GEL app can also be used to identify changes in protein band sizes due to post-translational modifications such as phosphorylation and so on. For example, you might know that the P53 protein is actually not 53 kilodalton on the gel, and that's because of many other factors. Such changes can be easily detected using GEL app and even help to identify possible disease-causing states. This is a video on how GEL app can be used, but out of time again, we will skip this. GEL app's accuracy comes from the measurement of the distance from the wells to the marker's location, and then taking the location of the sample to try and estimate the band size. It works for both DNA and protein gels, as you can see here. We have published an article on it trying to use Monte Carlo tree search to improve the accuracy of the GEL app quantification or auto-processing. First, we hand-labeled some of them with our expert eye, and then we trained the machine how to read both DNA and protein gels. For protein gel, it was easier because you could also incorporate color in the markers, which you can't do in the DNA gel. And from here, we were also able to rule out in case of a situation where the bottom bands of the marker run out of the gel, they will go by the best match. As you can see, the left and right on the right panel, the red and the purple are actually the same thing. So it will go for a best match for the marker, and then from there, it's easy to determine the size of the samples. We've even included making or a special wet lab method where without the use of vacuum, we can make better gels. So playing the video, you can see that we put a polyacrylamide gel in ethanol. Ethanol solves most of the problems in the world. It's just how much you have of it. And from here, you could see the polyacrylamide gel. This is a gradient gel, changes color, and allows a picture to be taken. Because of its shrinking and of course because of its turning into opaque white, it does allow faint bands to be found better. Okay, and this combined with gel app allows a better quantification of SDS page gels. And this is how it will be. It's a card size and it's reversible. Throw it back in plain distilled water and it will go back to the transparent gel again. Analysis showed that some of the faint bands that could not be detected by other methods can now be detected with this method. We have also used gel app to show and quantify these faint bands and automatically detect them much better than if they were not processed in the same manner. The last app I'm going to share with you is the colony counting app. There's two versions of it. There's the Lite and the Pro version. This app came from a need. We did not have a colony counter, and we were doing an experiment where we were trying to determine the best methods for producing best competent E. coli cells. And it was preliminarily done by an intern counting the colonies manually. And the numbers are sometimes very high. And so to replace this, which we didn't even have a colony counter. And even with this colony counter, some of the old ones, you still need to dot on them for the device to register. But with the smartphone now and so on, you can easily take a picture and auto-process them. So there's no more need to count colonies. Again, instead of having students or staff queuing for the colony counter, everyone has one in their phone next to their gel app. To separate the connected colonies, we use the watershed algorithm to distinguish colonies that are merged together so that we do not undercount these colonies. And the Lite version, you do have to tap on your phone as you would on a colony counter, but the Pro version does automated processing where it automatically detects. With this, we've come to the end of the presentation. I would like to thank the staff and interns past and present that helped develop the devices and the apps. Certainly, I hope we've shown that it is possible to move the labs towards a mobile phone-based lab, and that when working from home in lockdowns means really working from home for the scientists as well, where great discoveries that were done by the great scientists of old in their own homes can now again be present in the modern day scientists. With our lab techs that can be folded into a phone, we can do much more. I'd like to thank Merck for this opportunity, and I hope to work with some of you and hopefully develop some of these products further. Thank you very much.